she's going to tell us about cytoskeletal mechanisms controlling embryonic neural crest migration. Looking forward to it.
Thank you. So I'm going to talk about something very different than the previous talks. So what I'm interested in is to trying to understand how cell migration is regulated in a living organism. And I hope that I can convince you that this is a very important question because during embryonic development, different group of cells has to move tremendously and dramatically, and then they will relocate into the different regions of the embryo and give rise to different tissue and structures in an embryo. So here are an example. So this is the development of a frog embryo. You can see it starts from a hollow sphere and it rearranges itself extensively to form a 3-germ layered embryo with the axis established. And now you can see the flat neural plate is folding to form a neural tube, and the neural crest cells are migrating laterally away from the neural tube. And here's another example showing the development of a chick embryo. So it starts from a flat disk of cells on top of the yolk, and the cells undergo tremendous amount of cell movements. And now you can see the shape is getting there, and the head is forming here, and the neural tube is closing in an anterior to posterior sequence. So I hope that you can appreciate how much the embryos has been shaped by tissue rearrangements and cell migration.
Excuse me, what's the rough time scale for this process?
For this, it takes about a day to a day and a half. Chick is a bit slower, so it takes about 2 to 3 days to get to this stage. Yeah, so those are actually pretty fast developing animals. So I want you to appreciate how much cell migration has been happening during those processes, and you have to notice that as cells travel in those embryos, the ground they are traveling on, which is the living embryo, is also undergoing extensive rearrangements and growth. So it is very critical if you're trying to understand how cell migration is regulated, it's very critical to study them in their natural context in the living organisms, because no in vitro structures can really mimic the dynamic environment inside the living embryo. So among the different highly migratory cell types, I choose the neural crest as a model to study how cell migration is regulated in the living embryo. So this is a mouse embryo showing with neural crest labeled in green. So you can see that the neural crest actually originates from the dorsal portion of the neural tube, and it migrates into different areas of an embryo to give rise to different structures. For example, in the head, neural crest cells will migrate into the craniofacial region and give rise to jaws and cartilage and bones in the face. They also migrate all the way to the heart to form some connective tissues there. They also migrate throughout the gut to form the enteric nervous system, and they also migrate all over the skin to form the pigment cells. So since neural crest cells give rise to all these different derivatives, they also any defects in neural crest development or migration will lead to lots of birth defects or diseases. So actually, about a third of all congenital birth defects are neural crest related. So among them, there are lots of craniofacial anomalies and some heart defects, as well as many others. And in adults, defects in neural crest development also give rise to several different types of tumor, including melanoma, glioblastoma, and neuroblastomas. So during the tumor metastasis, as well as during normal development, cell migration plays an important role during those neural crest lineages development, because the neural crest cells, they have to migrate along the correct pathway to the correct to be exposed to the correct signals that were given from the environment, and then to arrive at the correct destination, and also to differentiate into the correct structures. So, so this is just a video showing you how dynamic neural crest migration is regulated. So this showing neural crest cells from the head region of a chick embryo migrating away. So you can see that it migrates along different streams, and it's undergoing very dynamic movements. So most of the cells will migrate into the lateral, laterally from one side, but then there is also always a small portion of them migrate across the midline and migrate into the contralateral side. So my goal is to understand how this amazingly dynamic migratory behaviors of neural crest cells is regulated. And for this, so I basically trying to approach this question from two different angles. The first is trying to understand the most basic and fundamental machinery of cell migration, which is from the angle of the actin cytoskeleton machinery, because that's the really basic mechanism that controls cell migration. And I'll use the cranial neural crest cells, which is most abundant and more easily accessible to study this question. The second angle I focus is on the transcriptional control of neural crest cells and trying to understand how the cell fate decision and the path finding is linked. And I use the long-term migration of cardiac neural crest cells in this study. So today, I don't think I'll have time to cover the second topic, so I'll only focus on the first research direction to understand how actin cytoskeleton regulation is controlled in neural crest cell migration. So here is just a diagram showing many different actin cytoskeleton regulators that have involved in different processes of actin behaviors and processes. For example, there's ARP203 and many others who are involved in the branching of the actin filaments, and this is involved in laminipodia type of protrusion formation. And there is a lot of actin bundlers who is involved in bundling the actin filaments, which is also involved in the filopodia type of protrusions. There's also lots of other linkers and the linkers to the membranes, for example, which is involved in the focal adhesion formation. So lots of those actin regulators are actually ubiquitously expressed throughout different cell types. So I'm curious to find out whether there's any special actin regulators that were utilized by neural crest cells for them to achieve this, their extraordinary migratory ability comparing to some other cell types. So from a microarray screen, we've identified a few cytoskeleton remodelers that are highly expressed in neural crest cells comparing to other cell types. This includes a myosin 10, a caldesmon, and a Cdc42 effector protein 1. So I found that all three actin regulators are actually required in neural crest cell migration, although they display somewhat different activities. For example, the caldesmon is involved in the directionality of neural crest cell migration. And on the other hand, the myosin 10 seems to regulate neural crest migration through mediating cell-cell and cell matrix adhesions. So today, I'm only going to focus on this unpublished work of Cdc42 effector protein 1 and tell you a story about how CEP1 regulates neural crest cell migration. So there's a family of CEPs, and they are identified through a East2 hybrid system using a TC10 GTPase as a bait. They all contain a crypt domain, which means they can bind, which is called a Cdc42 or RAC interactive binding domain, suggesting that it can potentially bind to Cdc42 or RAC. So there are a total of 4 CEPs in the Xenopus embryos. So I first look at their expression. So you can see that comparing to Cdc42, which is ubiquitously expressed throughout the developing frog embryo, so those are all, embryos from late gastro and early neural, early tailbud and late tailbud stages. And these are in situ showing their RNA expression. So you can see Cdc42 is basically everywhere, but then different CEPs are expressed more or less specific in different tissues, suggesting that they may mediate different activities of Cdc42 in different tissues. And among them, CEP1 is highly expressed in neural crest cells. So like at early stages, it's expressed in the pre-migratory neural crest cells at the borders of the neural plate. And later, it also expressed in the migrating neural crest cells as they populate the branchial arches. And well, at later stages, also shows up in the somite. So I'm going to focus on my study on this gene. So there is not too much known about different SAP's functions in different organisms. However, there is a wealth of knowledge of Cdc42 in cell morphology and motility. So Cdc42 is most famous for its role in its ability in polarization. So showing in a diagram here, Cdc42 is enriched in the leading edge of a migrating cell, and its polarization itself will lead to the polarization of other tissues and structures, for example, actin filaments and microtubules, as well as a polarized distribution or secretion of many other proteins. And all this together leads to the formation of filopodia protrusions, the asymmetric cell division, as well as the establishment of apical basal polarity. So my plan is to find out whether CEP1 mediates some of these activities of Cdc42 during neurocrist cell migration. So first, I just target the CEP1 into the xenopus embryo. So xenopus embryos actually has a lots of advantages for this early embryonic manipulations. For one thing, the cells are very big, so that we can directly target RNA, morpholino, DNA into a cell of interest, and then also have a very well-defined fate map so that we can know, relatively cleanly target specific tissues. For example, at A cell stage, if we target one of the docile animal cells, they will mainly give rise to neurocrist tissue. So here is the result. So here control or CEP1 morpholino is injected into one of the cells marked by the star here. And then the expression of, or the development of neurocrist is assessed by the in situ of a neurocrist marker SOX10 in blue. So you can see that during the specification stages, CEP1 morpholino doesn't affect neurocrist specification. As you can see that the SOX10 expression is symmetric in the uninjected and injected site. However, during neurocrist migration, when CEP1 is knocked down, neurocrist cells don't migrate very far, and we won't be able to distinguish the different migratory streams. And this can be largely rescued when you add CEP1 back. So this suggests that CEP1 is required for neurocrist cell migration. So next, I want to find out whether CEP1 is required within neurocrist cells for their migration. So whether it's required in a cell autonomous manner. And to test this, I performed the grafting experiment where the cranial neurocrist explants from an injected embryo is grafted into an uninjected host embryo so that we can observe the behavior of those cells, and only these cells receives the morpholino. So this is the result. So you can see that the control grafts can nicely migrate it into the branchial arches and segregate into 3 to 4 migratory streams. In contrast, when CEP1 morpholino is expressed in those cells, they don't migrate very far, and they fail to segregate into many migratory streams. And this also can nicely rescued when you add CEP1 back. So this confirms that CEP1 is required inside the neurocrist cells itself for to regulate its migration. And we know that cranial neurocrist cells give rise to lots of downstream derivatives, such as the cranial cartilage and bone. So we next just look naturally whether there is a consequence in the cartilage formation. And apparently, so this is the Alcine blue staining for cartilage. And you can see that when CEP1 morpholino is CEP1 is knocked down in this half of the embryo, the cartilage formation, the cranial facial cartilage formation is largely abolished. So the next question I'm asking is, how does CEP1 regulate neurocrist cell migration? So we now see that the neurocrist migration is affected when CEP1 is knocked down. So how does it really happen? So I take advantage of neurocrist explant culture experiment. So now instead of grafting the explant back into an embryo, now I just plate it on a fibronectin-coated cover slide. So this approach actually provides at least 2 major advantages. One thing is now the cells are allowed to migrate on the flat cover slide so that we can capture high-resolution images. Well, on the other thing is this explants maintain these amazing properties of the cell migration that you can see that it highly resembles the endogenous migratory pattern. Although it's isolated onto in a cover slide, it can still segregate into several migratory groups. So, so that we can kind of still see how the cell behaves and trust the results more. So this is just an example. You can see the neural crest cells have already migrated into 2 migratory groups, and there are still cells joining one of the group, and the cells are migrating relative to each other within the migratory groups. So I basically recorded a similar movie now with the actin filaments labeled. So you can see this is in controls. The green is the actin filaments, and you can see that it flows around the cells very dynamically. So it's enriched in the leading edge, and as the leading edge reaches full extension, it rapidly relocates to new sites of, to new positions for the formation of new protrusions. So it's constantly floating around the cell in a very dynamic manner. In contrast, when CEP1 is reduced in those cells, so now I used a much lower dose of the morpholino so that the cell morphology or migration is not completely abolished, so that we can still see a difference. So you can see that now the actin filament basically formed rings around the cell cortex, and they move in a ripple-like manner. So the cells fail to form protrusions. So it seems that the actin either wasn't able to stabilize or polarized in those cells, and then they, as a result, won't be able to extend membrane protrusions. So for a summary of this part of the talk, the CEP1 is specifically utilized in neural crest cells, and it's required for cranial neural crest cell migration, and as a result, in a cranial cartilage formation. And it seems to regulate actin organization and actin dynamics. So the next question I want to ask is how CEP1 interact with Cdc42 during neural crest cell migration? So for example, whether Cdc42 and CEP1 interact with each other, and whether CEP1 helps Cdc42 establish their polarized distribution in a migrating cell. And if that's the case, whether a change of CEP1 level will lead to the change of the cellular distribution of Cdc42. So to answer these questions, I first want to find out whether CEP1 and Cdc42 can physically bind to each other, or whether they are located in the same regions to allow them to physically bind to each other. So I performed this co-localization experiment. So here, EGFP fusion Cdc42 and the Cherry fusion CEP1 are expressed at low levels into the neural crest cells. And you can see that Cdc42 is expressed in cell membrane as well as in membrane protrusions. CEP1 is expressed in a somewhat different manner, so it's also expressed at low levels into the nucleus, but it's also expressed in membrane protrusions in some actin bundles. So from the yellow color, you can see that they are partially overlapping, which allow them to have some interaction, like functional interactions at least. So next, look at how the levels of CEP1 changes the expression, changes the distribution of CEP1, Cdc42, sorry. So here, this is the control of Cdc42 by itself. You can see that it's actually, so this is the an Explant, and the cells are actually migrating towards the right. So you can see the cells form protrusions only on the right side, and Cdc42 is enriched in this membrane protrusions. When CEP1 is overexpressed in these cells, you see that the Cdc42 is actually enriched all around the cell. So you probably can see this better if I show the green channel alone. So, okay. So you can see the cells form protrusions in all directions, and Cdc42 is enriched in all different directions of the cell as well. However, when CEP1 is knocked down in those cells, you can see, first of all, the cells adopt a much rounder shape, and they don't form much protrusions. And Cdc42 is redistributed in a more diffuse manner, so it's expressed in the cell membrane all around the cell, as well as in the cytoplasm. So conversely, also look at how Cdc42 level affects the distribution of CEP1. So, so the red channel doesn't work as well as the green, so I have to forgive me for that, but I hope you can still get the message crossed. And also the red channel actually tends to picks up more autofluorescence. So you can see all those, you have to ignore all those tiny red dots, which are the autofluorescence from the yolk platelet in the cells. You can see that in controls, CEP1 is expressed like in the cytoplasm and also in, like, at the base of the membrane protrusions and occasionally in the cell membranes. But then when Cdc42 is overexpressed, CEP1 is fast to the cell membrane. So you can see by the yellow color that there's more CEP1 expressed now in the cell membrane. And this is the red channel alone. When you ignore the autofluorescence, the background dots, you can see that CEP1 now is highly expressed in the cell membranes. And in contrast, when Cdc42 is reduced, you can see that now CEP1 is somehow recruited back to the nucleus. So I don't really know what CEP1's role is in the nucleus, but it's less likely that it's playing a role in cell behaviors when it's in the nucleus. So for a summary of this part of the talk, Cdc42 and CEP1 express in an overlapping manner in membrane protrusions, and they do regulate the cellular distribution of each other. So this is a very crude model, so that in control conditions, Cdc42 and CEP1 are enriched in cell membranes and in membrane protrusions or close to the cell membrane, and they are distributed in a polarized manner. When their levels are increased, they are expressed in cell membranes or membrane protrusions all around the cell in multiple locations. And when their levels are reduced, cells don't form much protrusions, and their distributions are, they are basically redistributed with Cdc42 distributed in a more diffuse manner and CEP1 recruited back to the nucleus. So, so next I would like to find out how Cdc42 and CEP1 cooperate during neurocrest cell migration. And so that's basically what I'm planning to do next. And for this, I'm going to employ the FRET experiment. So the FRET is the technique to, based on the image energy transfer between fluorophores. So when the two fluorophores are close enough to each other, the excitation of the first one, such as the EGFP, will transfer energy to the second one, such as Cherry, and leads to its emission. So this way, I will be able to tell how whether the two proteins are interacting with each other during in the live cell during their migration, during their behavior. So I'm going to use the desensitized emission method, which is going to be very tricky and challenging. But if it works, the results is going to bring very important results, because the time and place of the interaction may correlate with the activities of their activities during actin organizations and during cell migration. Another thing I would like to do as well is to, because similar to Cdc42, CEP1 may also interact with REC1. So I'd like to use a similar approach to characterize the interactions between REC1 and CEP1. So with that, this is basically the big picture. So this is sort of my long-term goal. So this is a putative actin regulatory network that I built based on yeast and cell culture studies. So that, so I hypothesize that extracellular signals will first activate core actin regulators such as Cdc42, REC, or Rho, which will then activate different actin regulators that either directly binds to actin or indirectly binds to actin to regulate different actin processes. And so far, look at a few actin regulators. I would like to characterize them further and plus look at different the activities of other actin regulators in vivo. For example, there's a facin, which is an actin bundling protein, and cofilin, which is involved in actin disassembly, as well as some close to the plasma membrane, like claudin, drebrin, and connexin. They are all highly expressed in neurocrest cells and potentially play a role in neurocrest cell migration. So I would like to combining all of the knowledge together and then ultimately understand how different actin regulators cooperate to regulate neurocrest cell migration or cell migration in general. Okay, so with that, I would like to thank my postdoctoral mentor and my colleagues. Dr. Yinki helps with the microarray screen, and Dr. Max Azin, who helps with analyzing cardiac neurocrest cell migration in another project. And I would like to thank the funding agencies, IDCR and American Heart Association. Thank you, and I can take questions.